Hi and welcome back to freesciencelessons.co.uk. By the end of this video, you should be able to describe how plants can be cloned by taking cuttings and by tissue culture. Remember that clones are genetically identical individuals. Clones are produced in nature by asexual reproduction. We can see that in this spider plant. Humans have been cloning plants for many years, so in this video we're going to look at the two ways that this is done. Cloning plants has one big benefit. Because the clone is genetically identical to the original plant, we know exactly what the clone's characteristics will be, for example the colour of flowers. If we used seeds, which are produced by sexual reproduction, then all the offspring are different. We can clone plants quickly and cheaply simply by taking a cutting. We normally use hormone rooting powder to encourage the cutting to develop roots. So this is a genetically identical clone of the starter plant. Now taking cuttings works really well if we just want one or two clones. However, what if we need hundreds of clones of a plant? To do this, we use tissue culture. In tissue culture, we take a plant that we want to clone, and we divide the plant into hundreds of tiny pieces. Each of these pieces contains a small number of cells. So this shows a scientist splitting a plant into lots of small pieces. These tiny groups of cells are then incubated with plant hormones and eventually develop into fully grown clones. The conditions that we use for tissue culture must be sterile, as we must not introduce any microorganisms such as bacteria or fungi. Tissue culture is used to produce lots of identical plants cheaply, such as these plants for gardeners. Okay, so hopefully now you can describe how plants can be cloned by taking cuttings and by tissue culture.